The University of Wisconsin-Parkside College of Natural and Health Sciences proudly presents Science Night. Tonight's topic, the biology of maple syrup.
We're focused tonight on maple syrup, and that's the kind of thing I think Dave and I get a little twitch in our shoulder or our head when March comes around. The other great thing is it's a time of year when everyone's ready to get outside. It's starting to warm up, and so it's great to put your boots on and slog through the snow. Start to see some of the spring flowers in a few weeks to months come up. And so many of you are probably aware of the general process. I'm going to walk you through the general method initially. So as you collect the sap from sugar maple trees, it usually ranges between 0.5 and maybe 2 to 4 or 5% sucrose in that sap. It varies with the time of year, the particular tree, and seasonally. And usually it happens between the end of February and April. So March is prime time maple syrup collection, particularly in Wisconsin. As far as across the United States, Wisconsin tends to be later season collection, whereas in the Northeast, people start collecting usually a bit earlier, but it varies from season to season. So after you've collected the sap, usually you cook it down, and traditionally it's usually outside over an open fire. You may have a refreshing beverage or two, enjoy a company of some friends in the process. Your goal is to evaporate 98% of the water to concentrate what's left so that you have about 66 to 68% carbohydrates, which is mostly sucrose that's left. And that's what gives it that viscosity, that thickness, obviously the sweetness as well. And we measure it in percent weight per weight, or also in bricks. So 1% weight by weight versus weight by volume is equal to 1 bricks. And that term bricks is used for other things. For example, in wine production, they'll measure sucrose content in the grapes, in the juice from the grapes before they start doing fermentation of grapes. So that bricks is a common term you'll see. Now, once you've concentrated the syrup, so it's about 66 to 68%, you then just collect it in bottles. And if you collect it when it's over 190 degrees Fahrenheit, it sterilizes the bottle, sterilizes the lid. You don't have to do any additional thing. You don't have to boil it for 15 minutes, such as for the canning process. You just cap it, and you can store it at room temperature or preferably at 4 degrees. Once you've opened that bottle, then you should keep it at a refrigerator temperature so it doesn't go bad with fungus or bacteria on it. So it's a fairly easy process. Now, traditionally, if you're thinking romantically about the process of collecting maple syrup, you may be thinking of large maple trees with lots of metal buckets. Maybe there's a horse drawing a sleigh with a container that you pour the syrup in. Then, you go to a romantic cabin in the woods where you have a fire and lots of evaporation taking place. And this still happens. That's many of the sugar bushes, both in Wisconsin as well as in the Northeast. And a lot of times have open days where you can come and collect sap straight out of syrup as it's being concentrated, put it on some snow so you have sugar on snow. It's a great kind of treat. Sometimes you can use that partially cooked down sap to mix it with like bannock, and you can make kind of a sweet bread with it. So that's the romantic version of it, and it still happens, thankfully. But traditionally, or now, the more common way to collect it is using tubing or piping systems that are collected either through gravity flow or vacuum systems that will collect it in tanks. You use reverse osmosis systems, which with a membrane, it can take that sap from 2% sucrose up to maybe 8 or 20% without having to heat it, just through a reverse osmosis process. The advantage of that is now if you're at 20% sucrose, you only have to use about 1/3 of the amount of fuel to be able to finish it. And it saves cost. It allows you to produce a lot more at a lower cost. And then eventually you do finish with an evaporator system, and the larger ones are usually steam-driven evaporation systems. And when they're cranking, they can produce about 1 gallon of syrup in 20 seconds when they're really at high efficiency. So this is how most of the syrup that's collected are produced, is being produced in Wisconsin, other states, and also in Canada. Now, what's present in the sap, or what's present in the syrup? So most of what's in there is carbohydrates. And for sugar maple trees, and even for silver maple trees, the majority of that carbohydrate is sucrose. Depending upon the time of year, it'll vary between 88 and 99% sucrose, which we all like sucrose. That's the sweet flavor that we crave, and that gives it much of that flavor. The concentration will vary depending upon what time of the year. As it gets later season, there's less sucrose, and you get more fructose and glucose. And those that are chemists in the audience, you'll realize you can cleave sucrose, a disaccharide, into fructose and glucose. And what happens is, later in the season, the weather's warmer, when the sap's in the bucket, there's bacteria and some yeast, they are cleaving the sucrose into fructose and glucose. And that's called inverting. And the enzyme that does it is called invertase. And so later season, you tend to have a higher percentage of fructose glucose and a little bit lower sucrose. And what happens when you cook it down, you vaporize it, or you remove the water, the fructose and the glucose will caramelize, and that gives it a darker color. So that's why late season syrup is a darker color. Sometimes it's called grade B or very dark grade A. And it's because of the change in the sugar content. But still, most of that sugar is sucrose. Now, what gives it the maple flavor? And it's not completely clear. It's thought to be a mix of a specific combination of amino acids. They're fairly low concentration. A little bit of a compound called maple furanone, some compound called strawberry furanone, which, as you might guess, is in strawberry. That's where it was first described. And also some maltol, which, here's some of the chemical structures of sucrose, glucose, and fructose. This is the glucose moiety, and here's the fructose moiety. When it gets cleaved, it generates these two. Here's the maple furanone, the strawberry furanone, and the maltol. And maltol was first described from the malting process. When you grow up a little barley, it converts all that starch, which is a bunch of glucose molecules, into disaccharides with glucose, and that's called maltose. That's used for the brewing industry, but there's also some of that maltol present, and it has a bit of a flavor. So maple syrup is a combination of these, as well as the sweetness being driven mostly by the sucrose. Now, early and late season syrup has very different appearances and different flavors. Again, what's happening mostly is late season you have more of the inversion, so lower sucrose, higher glucose and fructose. So when you heat it, it caramelizes, and it's a bit hard to see, but you get some darker colored syrup. This is late season. Here's maybe some grade B. Here's early season, more of that amber, sometimes called fancy syrup. It's all grade A. It all has the same sucrose concentration, but it has different fructose-glucose combinations. Usually at the end of the season, what drives the stoppage, that is stopping collect sap, is when the syrup starts to have a twiggy or a budding flavor. What's happening is the trees are actually beginning to leaf out or getting close to leafing out, and there's other organics in the sap that give it a bit of an off flavor. And that's usually what defines the end of the season, growing season. And it varies from year to year. Depends upon when the cold and the warm weather change, and so it can change dramatically. Now, one of the things that's changed very recently is how the USDA grades different maple syrup. The newer grading system, it's going to be all grade A, labeled golden, amber, dark, or very dark, and then kind of the lower quality called the processing grade. This is used only for additives, say for food additives, for large production or commercial level. These can only, by law, be sold as 5-gallon buckets. But all of these would represent the grade A that you can buy through retail. And this is a very recent change. So as of January, USDA has changed to a new grading system. It frankly matches more of the international grading system. And consumers have kind of moved towards favoring the dark ambers, where maybe 20, 30 years ago, the fancy or the lighter ambers were preferred. And so here you can see the old system, which is still being used in the grocery stores now. Here's the newer grading system. All of these can be sold by retail, so in grocery stores. And this gives you a sense of the color variation. So the grade A golden is the lighter. It has more of a delicate flavor. The grade A amber, a little darker, but it has a rich flavor as it's described. Grade A dark has a robust flavor. And finally, grade A very dark has a strong taste. The grade A very dark used to be what was called the commercial grade. And so that could not be sold by retail. But now it's been made so it can be sold by retail. So that change is coming through right now. And so some of the producers in the middle of transitioning. My understanding is they have until 2016 to legally change. So it'll take a little while for that transition to take place. So these are the different grades that are being sold. Now, what about Wisconsin, syrup production in Wisconsin? So in Wisconsin in 2013, there were about 265,000 gallons of maple syrup, so finished syrup that were produced. That's about $10 million worth of product. The average sap content was 2%. That's statewide, at least in 2013. And Wisconsin makes about 6% of the maple syrup produced in the United States. Most of that, it's a bit hard to see this down here, but 62% of the Wisconsin maple syrup is sold bulk, sold for additives, for food, so it's not sold retail. More of it's sold for commercial production and for food additives. How about at the United States level? Well, first off, the U.S. only makes about 20% of all the maple syrup made worldwide. The other 80% comes from Canada, which is not surprising considering the maple leaf is obviously the flag. And most of that maple syrup in Canada is produced in Quebec, so the northeast portion. So it's about a $130 million industry in the United States as far as maple production. But again, most of the syrup produced in the world comes from Canada. So Canada kind of wins out. And that's part of the reason, again, there was a change in the grading so that it matches particularly the Canadian grade system. And it makes sense as far as being able to sell back and forth across the borders. This is also a graph. These data come from USDA reports, the most recent one. This is the production at a state level in the United States, and you can see Vermont wins. It makes the most maple syrup in the United States, about 42%. Maine and New York, they both tie about 17%. These are data from 2014. And Wisconsin has the fourth largest production in the United States at about 6% of the syrup made nationally. Okay, so that's a little bit about the chemistry and the production level of maple syrup in the United States. I do want to make a slight divergent and let you know that there's other trees that you can make syrup from. For example, birch is a fairly common one that people make birch syrup from. It's fairly expensive. I know Dave was looking it up the other day, and I think it's quite pricey. We thought about maybe buying a little bit of birch syrup, but I think we thought it may not be, may break our bank, so to speak. But you can make birch syrup. Now, looking at the chemistry of what's present in birch sap, it's a little different. And that's part of the reason birch syrup has a little different flavor, a little different texture. It also comes out about 0.5 to 2% carbohydrate in the sap, similar. Finished birch syrup is also 66%, so it matches maple syrup. But look at this. The difference is primarily in lower sucrose, higher fructose and glucose content. So when you evaporate it, when you concentrate it, what happens is these high fructose and glucose monosaccharides, they caramelize. And so you get this really dark, rich birch syrup. And it has almost a molasses-like texture and flavor. So it has a little different flavor and texture because of that difference in the carbohydrates. Okay, so I want to transition from kind of the production side and then talk a little bit more about what's happening in the trees, what's happening at the physiological level in the maple trees. So I want to kind of cover kind of two big questions in the last maybe 5 to 10 minutes. That is, where is the sucrose coming from in the tree? Second, why is it pressurized so that when you tap it, you drill holes and you put spiles in, the sap comes out? And thirdly, why spring? Why is that such a good time of year for collecting the sap? So, as I'm sure you all realize, all of that syrup and the sugar in the syrup is really coming from last season's photosynthesis. Both the high-energy compounds, the carbohydrates, but also all the carbon that's present in those carbohydrates. And both of those are necessary for the next growing season. Because as you know, in a deciduous tree, wintertime, the leaves have fallen. The only way they can make new leaves is by having that reserve energy and that reserve carbohydrate stored so that when springtime comes, there's energy and carbon to make more leaves, there's solar panels to therefore go through another season. So when you're eating maple syrup or having a food with it, keep in mind you are eating last summer's sun in essence, and the energy from the sun. So it's all being driven by photosynthesis. This is happening the season before. Now, and I'm not going to go through lots of biochemistry here, but if photosynthesis is using the light energy and carbon dioxide from the atmosphere, and in the chloroplast, those are the subcellular parts in the leaves where this process takes place, that carbon is fixed ultimately into carbohydrates that initially is usually stored in starch. In starch, it's just a long chain of a bunch of glucose molecules. So you put a bunch of glucose molecules together, and it's starch. In contrast, cellulose is also a bunch of glucose molecules, but with a slight different bond. And so we can metabolize starch, but we cannot metabolize cellulose. But starch is the most common way that the plants will store this high-energy carbohydrate and the energy from the sun. So starch is a common way. But this can be converted through several biochemical reactions into sucrose. So it chops up those long starch molecules into smaller sucrose, which is glucose and fructose. And then that can be transported out of the plant cell into the vascular tissues, into the phloem and the xylem that many of you would have learned about in probably middle school or high school or maybe younger biology classes. And then it's transported around the plants, both in plants but also in trees. Well, I guess trees are plants, but herbaceous plants as well as within the trees. Sucrose is the common sugar that is moved throughout the plant. Now, in the fall, when the tree has gone through a season of photosynthesis, it's collected all that energy, light energy, stored it in carbohydrates, most of it at that point is in the leaves. But the deciduous trees have a challenge. If they drop their leaves and they don't harvest out that carbohydrate, they've lost that season worth of photosynthesis, right? So what they do is they will pull that energy out. They will break down the starch into sucrose, transport it out of the leaves, down the branches, into the trunk, and most of it, late fall, is going to be stored in the roots. So it's where that energy and the carbon is going to be stored to overwinter. And so this transport is happening from the leaves, through the petioles, branches, trunks, and down eventually until most of it ends up in the roots. And the goal is before the leaves turn completely and they drop to get as much of that energy and the nutrients back out before they lose their leaves. So the process is we have this sucrose that's being transported down the branches, the trunks, into the roots. Once it gets into the roots and into the trunk, for the most part, it's going to be stored in those cells back into the form of starch. And usually that happens in these structures called idioplasts or amyloplasts. And you may or may not know it, but you've eaten a lot of amyloplasts. If you've ever had a potato, what you're eating is plast is chock full of starch, and that's what amyloplasts are. And those are present in the roots and the trunks cells in those regions. Again, starch is just a bunch of glucose molecules connected. So this is what's happening late fall, early winter, before they lose the leaves. So the challenge is if they don't do it, they're going to lose all the leaves, and they've lost that entire year's worth or most of that energy that would have been stored. So when it comes late winter, early spring, the reverse is going to take place. So now it's going to mobilize that energy, carbohydrates, move it back up through the tree, through the trunk, into the branches, up eventually to the buds, where the new leaves are going to start forming in the spring. So this reaction goes in reverse. Starch is converted back into sucrose. It is then transported out of those root cells and those trunk cells into the vascular tissues, the phloem, and also into the xylem, the sapwood, which is a bit unusual to carry carbohydrate in xylem, but in the spring especially that's happening. And so all that sucrose is going to be transported back up late winter, early spring. So eventually it's going to be in the leaf buds where they're going to start producing new leaves for the next round, next season of photosynthesis. So it's a controlled process. In plants, the trees are sensing the day length, they're sensing the temperature, they're measuring how long the days are to control, to regulate this on a seasonal basis. Now, that's kind of what's happening seasonally. But the other question I posed was, why is there movement? What's causing that movement of sucrose from the roots back up to the branches? When they don't have leaves for transpiration, that's a big question. Well, it comes down to chemistry for the most part. That is, and this is a classic experiment, if you have a high sucrose concentration, say in a cell or in this thistle tube for this experiment, and outside of it you have pure water, the process of diffusion or osmosis across the membrane is going to drive water moving into that high sucrose concentration, and that's going to create pressure inside of that cell or inside of this thistle tube. Well, let's think about that. What's happening at a cellular level? So here's a diagram, although notice my roots are on the top and the buds are on the bottom, so obviously this is inverted. But in the roots, if you have starch that's being converted to sucrose, sucrose through an active process is being dumped into the vascular tissues, and then you have that osmosis taking place. Water's being pulled from other cells, being pulled out of the soil, under the frozen ground, into the roots, and that's going to drive movement of water into these cells. Well, it's going to pressurize it. And the pressure ends up being 70 to 80 to 100 psi. So it's a significant amount of pressure that's forming inside of these roots. Well, if you get further up the tree, I know my diagram's inverted, but on the top of the tree where the buds are, it's a lower sucrose for several reasons. One, that's not where the starch is stored, and two, sucrose is actively being pulled out of the vascular tissue into the cells that are going to start leaf production. And that's going to reduce the pressure. So you have high pressure in the roots, low pressure up by the branches, and that drives what we call bulk flow. And then that extra water gets cycled back down, and it creates this cyclic, this process of bulk flow, And that's what's driving movement, for the most part, out of the roots, through the trunk, up to the branches, where that's where the leaves are going to form. So what do we do when we're tapping the trees? We're just drilling a hole in the vascular tissue, sticking a spile in there, and collecting some of this sap that we then cook down into syrup that we enjoy so much. So that's really what's happening inside of the tree. Or that's the best prediction. I mean, that's based upon a lot of good models. There could be other things. There could be root pressure that's adding to that, but those are some of the main forces. The last part I want to talk about is why spring? What's going on to drive that process? And it also relates to the physiology as well as the seasonal difference. So the tree is sensing that the days are getting longer, temperatures are getting warmer. Those changes in day cycles are inducing gene expression, so those enzymes become more active. When the temperature warms up, those proteins and enzymes become more active. And without photosynthesis, the way the cells get energy is they respire. Just like you and I respire, we breathe in oxygen, and then we use that to a carbon that we have to generate energy. Plants are doing the same thing. They don't have lungs, but oxygen is getting into those cells and tissues. But the product of respiration is CO2. Under cool temperatures, CO2 will dissolve in liquid. But as more CO2 is produced from respiration, some of that starts to concentrate, and when the liquids warm up, they have the lowered ability to hold CO2 in liquid, so it comes out of solution in small bubbles. And then, as you also know, air will expand when it warms up. So the thought is, between osmosis, the sugar concentration, the CO2 that's being released because of respiration, the pressure that those gas bubbles are forming, that adds even more reason to have pressurize, or more explanation for the pressurization. So in the spring, you have the right combination of longer days, warming temperature that's driving these metabolic processes that the tree is responding to generate this osmotic pressure, CO2 gas, and that's driving this bulk flow. And again, what we take advantage of is we put a tap in there and we collect the sap so that then we can enjoy it, or if you're a kid, you just, maybe you don't have to be a kid to do this. You stick your mouth underneath the tap and you get a little bit of sap. And in fact, you can usually taste a little hint of sugar if it's 2, 3, or 4% sucrose. Okay, so that's where I'm going to stop and transition, and Dave Rogers will take over. And at the end, we will have a chance to sample if you're interested. Come on up, Dave.
Well, thanks, Dave. That was great. I learned a lot. So I'm going to talk a little less hard science and more about a little bit of ecology. So first, I thought I'd start out with how to identify the plant itself, and then a little bit about some of the ecology of sugar maple today and the changes that are happening related to climate. And then I'll talk about the UWP Sustainable Landscape Initiative and how we're trying to produce maple sugar as well as lots of other products here on campus as part of the integrated classroom. I start off with this as our transition slide just to recognize that this process was learned by American settlers from the people that were here first. And I always love these old photos. You can see handmade birch bark buckets, handmade spiles, and they would have boiled it with a variety of methods, typically hot rocks, because obviously the birch would burn. So it's an old process that we're reinvigorating here. So here's the basic biology. If you have any trees in your yard and you're trying to figure out whether you can tap them or not, the good news is that most maples contain sucrose and have flow in the springtime, and so it can be used to make sugar. The one exception being the Norway maple, which unfortunately is the most similar to the sugar maple, which is the highest sucrose concentration species and the most desirable for sugar production. You can see they're quite similar. What you're really looking for is these, they tend to have more lobes, and then they have these very acuminate teeth, meaning they taper, they take a long time to taper, and so they have like very short, sharp, pointy looking teeth, where these are less so, and there's just generally a smoother appearance. Norway maples also get this black fungal spot in the fall. So if your trees get that, then it's a Norway maple, and you probably want to avoid it. Norway maples have this milky sap that's quite bitter and so is not suitable for sugar production. Over here is silver maple. It's quite different leaf-wise, deeply incised sinuses, much more jaggedy in appearance. But those, you can tap those. If that's all you have, they make a pretty good syrup as well, so they're quite suitable. But you're really looking for these sugar maples, Acer saccharum. But you know, this time of year, knowing what the leaves look like is sort of pointless because there are no leaves on the trees. They being deciduous. So I made sure I included the real deal here. So you got to look at the twigs if you really want to tell a sugar maple from the others. And you can see both Norway maple and the silver maple have fewer bud scales, right? And they're rounded. And these tend to be red, and this is a little bit washed out, but it has a redder twig here in the Norway maple. And they're also quite a bit larger than the other two species in terms of the overall size. Silver maple, they have fewer bud scales as well, but they're a little bit smaller than you'd see in the Norway maple. The sugar maple is quite distinct, where they're quite long, and they have maybe 11 to 13 scales, as well as these two sort of ancillary scales, and it's actually hard and pointy, like it's pretty stiff, so it's pretty distinctive. The look of these in the bud form, and so when you see in that, that's what you're looking for. You can also look at some bark characteristics. The Norway maple tends to have a thinner bark, and it has these showed up pretty good, these sort of light-colored valleys in between the ridges on the bark. That's, especially when they're young, that's very distinctive for this tree. And it's also pretty obvious as they get older too. So that's kind of what you're looking for. Silver maples tend to have multiple trunks and to have peeling bark. So they have like shredding bark. It almost looks like a cat scratched it or something like that. Whereas sugar maple has ridges and valleys, but it's all this lovely light gray color, and it's quite a stately tree too, where Norway maple, I'll just say it is ugly. It's an ugly tree. I hate it. I do. Where sugar maple is a beautiful tree. I don't know why anybody would plant something else. You got to be a little bit careful with your sugar maples because they actually go through quite a bit of a transition in bark texture. The younger ones are smooth. They almost look like a basswood. As they get older, they can kind of look like maybe like a red maple or something like that. And then as they get older still, they have that really characteristic sort of deep furrows, right? So that's how you can tell whether you have a sugar maple or not in your yard or in your woodlot. Sugar maple has pretty broadly distributed. This is a little bit exaggerated. This is like the known range, so where you could possibly find a specimen. But really it's in the more northern latitudes where it would be a dominant tree traditionally. And you can see, it's restricted here to the northeastern part of the U.S. Often, people who look at plant distributions and plant geography are really evaluating climate barriers, assuming that climate is the limiting factor that controls distribution of species, right? So you can see they'll look at the northern limit and the southern limits, look at the average temperature in January or July, and try to imagine why that limits that particular species in the sort of climate envelope, right? Interestingly enough, I just recalled this minutes before I came down to give the talk, that sugar maple, as well as ash and some of our other common trees, actually have a disjunct populations in Mexico and parts of Guatemala too. So you'll find these like little sugar maple forests up in the cloud forests of Mexico and Guatemala, right? But they're very distantly related, but it's the same species. They're deciduous. I would imagine they make syrup. They could. But they're not really well described, so that's just an interesting little ecological tidbit I'll throw out there. So with climate change is a big concern for producers. You saw the dollar figures that are attached to the economics of maple sugar can be quite substantial, and there's people whose livelihoods depend, at least in part, on maple syrup production. So like many things, there's a lot of concern about the current ongoing changes in our climate are going to, what impact that's going to have on our economies and in biodiversity. So you can see here several 5 different models predicting future climate scenarios. And essentially what they do is they build those same climate envelopes with the predicted climate where we see sugar maple today, and they predict where they would see it, where that envelope exists in these projections into the future. And then you can get these range maps, and you can see out of the 5 different models that were run in this particular instance, only one of them actually retained sugar maple in Wisconsin, and the others don't even have it anywhere in the US. It goes all the way up to like New Brunswick or something like that, very far away, right? So there's a lot of concern, not just in Wisconsin and the US, but also Canada, that this could be disappearing, right? And it's one of the species at risk, is sort of a poster child for climate change. And you can see what that future would hold. I personally just love maple syrup, and would be very sad not to have it. But meanwhile, back in the real world, I like to say, I have a paraphrase here from Mark Twain: "The report of my death was an exaggeration." If you look at the actual long-term data, not just here in Wisconsin, but throughout the Northeast, you're actually seeing, so these are data I collected as part of my dissertation. In the 1950s, this is the total number of stems that we observed in 128 plots, that were observed in 128 plots across southern Wisconsin. And this is the same plots revisited with the same sample size in 2004, and you can see that the number of maple stems has skyrocketed. And you're seeing this all over the northeastern US and all over forests in Wisconsin, that sugar maple is doing great, really. I mean, there is so much of it, and it's growing like crazy. Why would that be? Well, maybe the climate model isn't the only factor to consider when looking at the distribution of plants. And what we're seeing is, again, this conversion of what we had here in the 50s and really at the turn of the 20th century. This is shagbark hickory, and white oak would have been and walnut would have been the type of forest that we would have seen around campus. And we're seeing a wholesale conversion to basswood and associate, but really sugar maple becoming the dominant tree. And we've actually resurveyed some of those since 2000, and it's even more dominant, to the fact we're not seeing anything else in the understory except sugar maple and sugar maple seedlings. One thing about sugar maple is it loves shade. It's the most shade tolerant of all of our woody plants, bar none. That and beech, but really sugar maple can live in its own shade, right? And it'll outcompete other plants for that same light. So it's like the climax species in the forest, meaning that it can replace itself. So once you get a sugar maple stand, it just replicates itself over and over again, right? And this entire understory is sugar maple seedlings, right? So we don't have to worry, at least in the immediate future, about sugar maple. And what's driving that is that our landscape that was dominated by oak and hickory used to have a lot of fire on it. The same woman that was collecting the sap in the birch bark baskets, her people were burning that landscape to promote good grass growth, to promote game, to clear areas for agriculture. And that persisted until the early part of the 20th century, when fires were suppressed. And really it's taken 100, 120 years for sugar maples to just really take advantage of that. So if they've been burning, they're very thin-barked, have very intolerant to fire. Since fires have been removed from the landscape, sugar maple is just increasing. So it wasn't just climate, but it was also fire and disturbance regimes that were really influencing that distribution of sugar maple. And the removal of the disturbance is what's allowing it to take over. Okay, that brings us to this idea, the UWP Sustainable Landscape Initiative. And I say initiative realistically in that, like, it's pretty much my initiative, mine and my students' initiative. In fact, a lot of administrators might be seeing it for the first time right now. I did present this to the all group about a year ago. And this idea really came out of my first year here. I'm in my 7th year, where I taught a class in restoration ecology, and the students were very engaged, right? And we see all this potential with our property. And like a good restoration ecology, we started to dig into the history of the campus and really realized that we have just an incredible opportunity here on the Parkside campus, given the 700 acres that we have. Yeah, so this is what we're calling the UWP Sustainable Landscape Initiative. And it actually goes back to the early development of the campus. So in the late 60s and early 70s, when the campus was developed, this is the original master landscape plan. And I have to say that actually have a master's degree in landscape architecture as well, so I've studied this. This is extremely innovative. When they built this campus, it was visionary, from a landscape architecture perspective. Where the whole idea I've highlighted here, that the natural landscape is to provide an excellent outdoor laboratory for the natural sciences. So any restoration class will, of course, dig into the history and try to understand, what has happened to this land over centuries. So when we found this, what a perfect synergy for this class and for the sort of the reinvigoration of this idea. So you can see it right in our history. And here's Dr. Wolf from geography taking advantage of just the sort of remnants that we have around us from this original design. And through that effort, presenting it to the chancellor and working with the administration, we have successfully started to integrate this into our planning. Instead of sort of haphazardly just trying to do it on the sly, we've recognized these areas as legitimate outdoor laboratories and started to integrate them into our planning documents, right? So that's a big first step. Rather than thinking of them as open space or as wildlife habitat, that we think of them as outdoor laboratories, just the same way we think about chemistry laboratories and everything else, and we plan for it from a resource perspective. So this is a shot from the most recent master plan, and you can see the outdoor laboratory turn being used here for the woods right behind Wiley Hall, as well as some prairies that we're going to develop and the like, as well as some of the remnants of the old growth, right? So I'm really happy with this progress that we've made. And the guiding principles are quite simple, because all you have to do is look at the mission of a university, and there's your guiding principles. And I've sort of simplified what you would see on our website, and simplified them how I'm evaluated, essentially, when I go up for my yearly evaluation: education, research, and service. So we try to build all three of those components into the landscape as we develop it. So education, we're trying to develop major plant communities, these sugar maple forests, restore some oak forests, some prairies, so when we take students out, we can show them examples without having to get on a bus. And also teaching gardens, so bringing these closer to campus, where we have labeled plants and labeled walkways, so we can educate not just our students, but also visitors to campus. Research is a really important component, and essentially restorations as experiments. It's an old saw among ecologists that restoration is the ultimate test of ecological theory. So, if we can understand how ecosystems work, can we reconstruct them? That's sort of the ultimate test. And then service, the physical environment for recreation. There's a ton of research now showing that exposure to natural areas, especially diverse natural areas, reduce stress, aid in retention for students, and all around are good for you, right? There's a lot of scientific evidence supporting that. So try to enhance that physical environment. And of course, to demonstrate sustainable landscaping, right, for visitors to the campus, and try to provide that sort of expertise to our collaborators across the communities, different communities. All right, so this is our first year was last year about this time. I think it was March 18th. So this is the first tree being tapped here by myself and Dr. Higgs, using the woods just outside of Greenquist Hall, the Greenquist Woods. And there it is, our first few drops of that sweet liquid. There's a spile right there, the type that we've used, that we're about to insert in the tree. And then here are the bags hanging. We did about 55 taps in 35 trees about. We have these hangers that you can see the spile right there. We hang them on there, the sap drips into the bag, and then we harvest the bag, fill up the buckets, put it in the gator, haul it over to where the cooker is, which is by facilities last year, and then sort of store it up in big barrels until we get around to cooking it, usually at the end of the week. Here's a shot of Vince. There's Vince cooking. Vince was amazing in that he put together the cooker, as well as put it on this nifty little cart so we can move it around to different places. So there he is stoking the fire. And there it is boiling away, as well as he rigged up this really nice storage tank so we can run it for hours without having to constantly monitor it, where we can actually kick back and relax and enjoy ourselves. So we have started to use the tapping as a resource for our classes. So this was with Dr. Higgs' class in plant physiology last year, where students get experience, collecting data, being organized with data, making measurements, and the like. So we had 37 trees, 57 taps. We had about average volume per tree, I don't know if that's per day, was about a quart. Right? This was sort of just a test case. Some instruments, this is how we figure out the concentration, the refractometer on any given, from the sap, loading it up, taking the measurements, and of course here's the end product. Right? The first year, of course, is just sort of a, how does this work? We hope to extend this, certainly starting tomorrow. Actually, we're going to tap tomorrow for my botany class. To look at some questions. One of the surprising things I found was that not every tap on the same tree had the same sugar content, but it actually varied quite a bit. So that's an area that we can investigate a little bit. Other things are like the health of the tree. Does the growth from the previous year or its light environment influence sap production, sugar concentration, and the like. Right. Here's some of the data produced. Not very compelling, maybe the first year around. It does show a pretty interesting shift from the mid-season to the late season in terms of the sap volume, as well as an increase, or at least some changes, in the sucrose concentration of the sap itself. So just a little bit of the sort of data that we hope to collect. And one of the great things about a university is we can think long term. So if we do this every year, over 20 years, we can accumulate a data set that normal researchers don't have access to, right? Because we can afford to just do it year by year. For example, these particularly low values, Dr. Higgs was able to tie to the fact that that particular tree number 31, is dying, right? It was too close to the Rita when they built it, and some of the roots got pruned. And you can see why it was, so really making that connection between the previous year's growth and the sugar production the following year. So those are the sort of things that we can do with these outdoor labs. It's also really great. One of the best things about this whole idea is that it's a way to bring people together outside of the classroom. Usually it's a very structured, and rightly so, student-teacher environment. But when we do projects like this, it allows you to interact with people in a more casual setting and to talk about things in that context when there's not a grade necessarily riding on the outcome. So you have students, we have staff, we have professors, a lot of times even the facilities guys, which usually you just see mowing and plowing, they come around and are very helpful. So it's a really good community building exercise. That's one of the most gratifying parts of this whole outdoor lab concept. And we hope this year, last year was a little bit we weren't quite ready to use the maple syrup production in particular as a way to generate revenue. Not, to support the university entirely, but to support clubs, right? We're talking about maybe a couple $1,000s a year, though, is what we're aiming for. 20 gallons. We got 9 gallons last year, 20 gallons this year. But, we can produce things, further refine the sugar into candies or maple cream, or these little maple sticks, which are a good way to sort of add value to the product. And then hopefully we'll be able to maybe tap into like a pancake breakfast fundraiser, maybe around graduation time when parents are here. Who knows? We could go anywhere with this, but the main thing is producing the syrup first. And we actually mocked up this nice label last year. Here's the Greenquist Gold, 100% pure maple syrup. So it's pretty fun. And this whole outdoor lab concept really has a lot of potential. This conversion to sugar maple is really as I said, a nationwide problem, and there's a lot of effort going into understanding it and how do we manage this. One of the problems is that you can see there's a lot of understory plants on the ground here. So this is really good habitat for birds and the like, where you get this very shady understory and you don't. It becomes just sugar maple seedlings, right? And part of the problem is that they're all isolated from each other. If they were connected to a bigger forest patch, there would be shade-tolerant plants that could invade those new shady forests. But because the nearest maple forest is, a long way away, those plants can't get in there. So what we need to do is add the plants or do experiments as to how we can encourage the understory to recover to be more like a natural maple forest instead of these new maple forests. There's records from Wiley Woods or Calmart's Woods that had this Ameriorchis in it, so it had orchids in it, and really over 100 species of vascular plants. And that was 25 years ago. When you look in there today, you can see this bare ground. You have all these young sugar maples and a lot of bare ground all over in there. So there's really either bare ground or sugar maple seedlings. So this is a management problem. That's not just on campus. This is region-wide. And so anything we can discover here will be eagerly utilized by other natural areas. So this is really important research, at least in my discipline. These are the kind of plants that have disappeared that were on that list that we'd like to bring back. And thankfully, our dean has invested in some seeds, and so this spring we're going to implement some of the initial demonstration restoration efforts. So that's good. We're just stratifying the seeds now. So we'll do seed addition experiments, and I'm actually part of a network of forest change that's international. And we'll establish some of these sort of global warming experiments too. So these are artificial warming chambers to see how climate change will play into this sort of this forest restoration idea. This is a newer component. So we have a lot of wood resources on campus. So we're growing, this is a new student that just started in my lab. We're working on growing out, these are shiitake mushrooms, which we grow it on plates. We purchase the cultures, then grow them out on plates, then inoculate these little wooden dowels, drill holes in, these are sugar maple logs. People typically use oak, but maples, and there's strains that are better for maples to produce mushrooms, right? So sort of a small scale economic production, but these are quite valuable and delicious. So that's another project that we've got underway, kind of under the same idea of the outdoor labs. And one that I'm really excited about is that this wood is extremely valuable, right? And I'm not talking about using it as like a timber operation, but working with Trenton Baylor in the art department, who is he's an excellent furniture maker. So we've talked about, carefully harvesting trees that are maybe declining or shading trees we want to save, doing a small scale milling operation, putting together a solar kiln here, and then using that to build furniture, right? And integrating that into the classroom, you could really marry science and art together, right? Because you could talk about trees and about ecology and about wood and wood formation, and then go out and select a tree, and then go through the whole process of creating art out of it, and really a holistic approach. And that's the sort of thing that I think these outdoor labs would really be excellent for. And again, sort of getting that interdisciplinary perspective, bringing people together rather than in their so-called silos. Right? So that's the vision we got for the outdoor labs, sort of beyond the maple syrup. But of course, we're here for the maple syrup. So, so that's all I had, I guess. End it. We'll take questions if there are any.